Alright, so now we're going to have a look at this new concept of phylogeny, or phylogeny, any way you want to say it. Essentially, we're looking at basically what are the methodologies in which we can compare the genetic relationship between two individuals to sort of see how their genomes compare to each other. So we're really diving into this idea of comparative genomics. So looking at us to see how closely related we are, and then also be able to infer then when was the point in time in which we diverted into two distinct species. For success for this lesson, what it looks like is that you're able to describe those three techniques that you see in the success criteria. So how we can actually use amino acid sequencing, or the sequencing of common proteins, to determine genetic relationships, DNA hybridization, or just DNA sequencing, which is something we've already learned about in the past. So taking a step back before we look at that though, is before we knew anything about this concept of DNA, and knew that all living organisms had these common features, we had scientists like Charles Darwin that were able to compare commonalities in sort of anatomical features or physical things that organisms share between them. And so he made some really key notes on, while there were similar structures, so homologous structures we call them, meaning that if he observed similar structures, ones that were the same, that there might be a similar point of origin of those structures if we went back and back from successive generations. So Darwin noted that, whilst things like some organisms had arms and others had legs and things like that, that there were similarities in some of the bones that existed there. So maybe there was different ways that they had a different function that evolved over time. And so that was one of the really key things back in those days, before we understood really the basis of DNA. But once we started to learn about DNA, we could actually look at things like similar bone structures, and although they serve a different function, we can go, oh, they must have a lot of the same genetic information, and we can prove that now. And so when you look at images like this is where we can start to really infer that sort of genetic relationships between different species, and we can look at different points in time where maybe similar structures might have evolved a different function and allowed organisms to exist in a different way. And after certain periods of time, they became distinctly enough different that we were then able to classify them as different species. And the way that we compare species is sort of via this concept of evolution. And so we look at comparative genomics, so a study in where we compare the genomes of organisms, and we look at basically how closely related the DNA sequences are between one organism and another. And so we can see here where we go, oh, if we look at that little picture there in the bottom corner, the human DNA is about 84% identical, and 84% identical to the mouse and the chicken, and 81% to the zebrafish. When we look at then similar things as well, there's another sort of 8% that exists on top of that. If you look at the comparison between a mouse, it's quite similar. So what that means for us is that we pretty much must be able to do a lot of the same things that mice can do, meaning that we probably have a lot of the same amino acid sequences, a lot of the same proteins, and a lot of the same polypeptides that are created subsequently. And so that's really important to know, because then that sort of gives us indication of, we are much closer related to mice than we are, say, a horse or a fruit fly, for example, probably more so the fruit fly, meaning that we've evolved probably more distinctly away from the fruit fly than what we did the mouse. So the mouse, we are probably have diverged away and became new species in a much later time period than the fruit fly. All right. So now we get into the three different ways that we can actually look at how we compare genomes. And we're going to look at the first one now, which is that concept of amino acid sequencing. So the primary structure of proteins, which you'll learn about in topic 1, allows us to observe the different amino acids and how they're joined in polypeptides and then eventually in proteins. And what we notice is that closely related species will have a lot of those amino acid sequences in exactly the same organization, because they make exactly the same proteins. And that is the really basis argument for amino acid sequencing to determine genetic relationships.